is this particular bacteria? Well, it is a gram-positive, catalase-positive, and a coagulase-positive bacterium that is a common pathogen in humans. It's called Staphylococcus aureus because it belongs to the Staphylococcus genus, which means it is part of the larger family of Staphylococcae. The Staphylococcus aureus is often found as part of the normal flora of the skin, the mucous membranes, and also in the nasopharynx. However, it can cause a variety of infections, which can range from mild to life-threatening forms. Here is a picture of the Staphylococcus aureus for your reference. So let's talk about its microbiological characteristics, starting with its morphology. So, the Staphylococcus aureus will appear as a spherical or cocci cells, and it is typically arranged in grape-like clusters, as we see over here. It is also going to stain purple, which means it is gram-positive in nature, and the size can range from approximately 0.5 to 1 micrometer in diameter. There are some useful biochemical tests we can do. So, the first one being the catalase-positive result. This means that the Staphylococcus aureus produces the enzyme called catalase, which will break down the hydrogen peroxide into your classic water and oxygen. If the test results indicate a coagulase positive result, that can also indicate a Staphylococcus aureus infection because this bacteria can also produce coagulase, which is an enzyme that converts your fibrinogen into fibrin, which would then lead to clot formation. This is necessary for helping the bacteria to evade phagocytosis. Finally, we have fermentation. The Staphylococcus aureus ferments mannitol, which can be detected using the mannitol salt agar. And so growth on the mannitol salt agar results in the yellow colonies because of the acid production from the mannitol fermentation. In terms of the grow conditions, the Staphylococcus aureus is a facultative anaerobe, which means that it can grow in both the aerobic and anaerobic conditions or environment. Its optimal growth temperature is 37 degrees Celsius, but be aware that it can survive in other temperatures out there. In terms of salt tolerance, the Staphylococcus aureus can grow in high salt concentrations, and that's why it can thrive on the skin and mucous membranes. When it comes to virulence factors, there are some key surface proteins, so they help the Staphylococcus aureus to adhere to the host tissues and evade immune responses. First one being the protein A. So it's a protein that binds to the Fc region of the antibodies, thereby preventing opsonization and phagocytosis. There's also this thing called a clumping factor, which will help to aid in binding to the fibrinogen and the extracellular matrix proteins, which then becomes necessary for bacterial colonization. We also have some enzymes, some of which we talked about earlier. So the coagulase is the enzyme that forms the fibrin clots, the fibrinogen around the bacteria. This prevents the phagocytosis of the Staphylococcus aureus, thereby allowing its survival. We also have the exfoliative toxin A or exfoliative toxin B. This causes skin peeling, which can lead to scalded skin syndrome. It causes skin peeling by disrupting the connection between the epidermal cells, which are also known as desmosomes. We also have enterotoxins. These are superantigens that can cause food poisoning by stimulating excessive T cell activation, and this results in cytokine release and inflammation. We also have toxic shock syndrome, toxin 1, or TSST1. The toxic shock syndrome, toxin 1, is a superantigen which will cause toxic shock syndrome by inducing widespread cytokine release and thereby inducing systematic inflammation. And then we have another thing called a biofilm formation. This is where the Staphylococcus aureus can form biofilms on medical devices and tissues, which would then make it resistant to antibiotics and immune responses. Now let's talk about the pathogenesis of Staphylococcus aureus. In terms of colonization, the Staphylococcus aureus colonizes the skin, the nasal passages, and mucous membranes without necessarily causing the disease. Approximately 20 to 30% of healthy individuals happen to be persistent carriers of the Staphylococcus aureus in the anterior nares. The bacteria can become invasive through the following methods. So first, the bacteria will enter the body through some broken skin, mucous membranes, or even through some implanted medical devices. And then we have infection. Specifically, the spectrum of diseases caused by Staphylococcus aureus includes folliculitis, impetigo, cellulitis, and abscesses. When it comes to skin and soft tissue infections, we can also have bone and joint infections such as osteomyelitis or septic arthritis. We can also have endocarditis, which can involve infection of the heart valves, and this commonly happens in intravenous drug users and patients with prosthetic valves. We also have pneumonia. This can be seen in immunocompromised patients or patients who previously had viral respiratory infections. We also have sepsis. So, unfortunately, this bacteria can cause systemic inflammatory response syndrome and ultimately septic shock. Remember when we talked about the toxic shock syndrome, toxin 1, which happens to be that superantigen that can cause the toxic shock syndrome by inducing widespread cytokine release and systemic inflammation. Well, this bacteria can cause this unfortunate circumstance, and the toxic shock syndrome can often be associated with menstruation, but it can also occur after surgery or skin wounds. We also have scalded skin syndrome, which is a condition that can affect neonates. This will be characterized by skin sloughing off due to exfoliative toxins. We also have septicemia, where Staphylococcus aureus can disseminate through the bloodstream, which can then lead to sepsis, endocarditis, or septic arthritis. Remember that septic arthritis had to do with some bone and joint infections. So, what is our body's response to the presence of Staphylococcus aureus, also known as health response? First and foremost, we have the innate immunity response, where the Staphylococcus aureus can invade or evade host immune defenses, including phagocytosis, and that does so through the production of capsules and surface proteins that would inhibit opsonization. We also have adaptive immunity, where the immunoglobulin G So, when we have used a lot of antibiotics, the bacteria has developed several forms of resistance that we need to be aware of. First, we have the methicillin-resistant Staphylococcus aureus, also known as MRSA. So, the MRSA, or this form of resistance, involves acquiring the MECA gene, which would then encode the penicillin-binding protein 2A. This will have a low affinity for beta-lactam antibiotics, thereby rendering them ineffective. It is clinically relevant because this form of resistance is associated with increased morbidity and mortality due to limited treatment options. It can cause both the hospital-acquired and community-acquired infections. So, to manage this, we may have to utilize vancomycin, daptomycin, or linezolid, and the choice depends on the severity of the infection along with the local resistance patterns. We also have a vancomycin-resistant strand, and this bacteria has developed resistance to vancomycin, and it did so through the acquisition of the VAN-A gene from the Enterococcus species, which would then result in altered peptidoglycan synthesis. And the treatment would involve using newer antibiotics like ceftaroline or daptomycin, even though these strains are rare. So how do we diagnose infection from Staphylococcus aureus? First, we need to do a specimen collection. So specimens may include blood cultures, wound swabs, sputum, urine samples, etc., depending on the infection site. And then the laboratory testing, we would first do a gram staining to see if we have gram-positive cocci or cocci clusters. We also do a culture where we grow the bacteria in agar media, such as a blood agar or MSA. And then we do a coagulase test, which happens to be a confirmatory test for Staphylococcus aureus. We can also do a molecular-based test, such as a polymerase chain reaction, which can help identify genes like the MECA gene for the methicillin-resistant strand, or toxins like the TSST1. So after we have confirmed diagnosis, we have some treatment options. So one common form is the antibiotic therapy. We can do an empiric treatment where for severe infections, we would use broad-spectrum antibiotics like vancomycin, ceftriaxone, or clindamycin, while we await culture results. Once we have the susceptibility results, we should then tailor the treatment to make it more targeted. So for non-MRSA infections, we can use beta-lactam antibiotics like nafcillin or oxacillin, but for methicillin-resistant forms of this bacteria, we need to use vancomycin or newer agents like linezolid, daptomycin, or ceftaroline. We can also do surgical intervention in some cases, where abscesses or infected joints may require surgical drainage in addition to the antibiotics. If we have severely infected tissue, a debridement may be necessary to remove the necrotic or damaged tissue. So, how do we prevent this particular bacteria from getting into our system and becoming infective? So we have some preventative measures. The first one being the hand hygiene. So remember that proper hand washing is the most effective way to prevent transmission. Screening and decolonization are also important. Nasal screening for Staphylococcus aureus in high-risk populations, such as healthcare workers, patients undergoing surgery, should be utilized, followed by decolonization with mupirocin nasal ointment and chlorhexidine washes. also lead to pneumonia, especially if patients have underlying conditions like influenza or cystic fibrosis. It can also lead to osteomyelitis and septic arthritis. And this can be particularly prevalent in children and those with prosthetic joints. Sometimes we can have endocarditis. It can often be acute in nature, and unfortunately, the mortality rate is high. Then we have the toxic shock syndrome, which we need to pay attention to, because this will present clinical characteristics of fever, hypotension, rash, and multi-organ involvement or impairment. And then the Scottish skin syndrome involves the blistering and peeling of the skin. Once again, it primarily affects neonates. So pay attention to these clinical signs and symptoms. Or if you find any of these, always consider a Staphylococcus aureus infection as one of your differential diagnoses. So in summary, the Staphylococcus aureus is a versatile and formidable pathogen, especially given its prevalence of antibiotic-resistant strains. So while it is often part of the normal microbiota, it can cause serious infections through its numerous virulence factors such as toxins, surface proteins, and enzymes. Furthermore, because of emerging antibiotic-resistant strains like methicillin-resistant Staphylococcus aureus and vancomycin-resistant Staphylococcus aureus, managing infections caused by this bacteria involves careful consideration of antibiotic resistance patterns, along with rapid diagnosis and appropriate treatment strategies. So, those are all the details of the Staphylococcus aureus that are important to keep in mind moving forward. Feel free to comment down below any other important information about this bacteria. Other than that, I hope you enjoyed this informational ASMR video, and I hope to be back with another one soon. Take care and peace out.